So in this video, I'm going to continue our discussion of the immune system. I'm really going to focus on the acquired immune system and how it deals with specific threats. So the challenge that we face is that oftentimes pathogens have co-evolved with us in order to avoid the non-specific defenses that I talked about in part 1 of the video. Just as one example, salmonella is a very nasty bacteria that we can ingest in food. So it's going through our digestive tube and it can survive the stomach acid that often kills other pathogens in our food. It can then make its way into our bloodstream, and there seems to be some evidence now that salmonella strains can even destroy the phagocytes that take it in through phagocytosis. It can sort of do things inside of those phagocytes that ultimately lead to their death as well. So a fantastic job by them, and a real unfortunate situation for us that they can really do some damage inside of us. So with these and other pathogens that maybe can get around our non-specific defenses, we're going to have to mount a specific attack on those pathogens with lymphocytes, the other type of white blood cells that I briefly identified in part 1. So for the lymphocytes to really attack a specific pathogen, they're going to need to be able to recognize them specifically. So how do lymphocytes do that? What they need to be able to do is find some kind of landmark on the surface of the bad guy. So any kind of landmark on the bad guy pathogen, whether that's a virus or a bacteria or a fungus or something else, any kind of marker on its surface that can be specifically identified as belonging to that pathogen, we're going to call an antigen. So the antigen might be on the surface of the bad guy. We're going to see that the antigen might be presented even by our own body cells when they're sick. But again, the antigen is some kind of surface marker on the bad guy. It might be some kind of membrane protein or membrane sugar, glycoprotein, or membrane lipid of some kind. Something recognizable to the bad guy. And the lymphocytes will have receptors that try and bind to that. And what I really want to introduce here early on is that basically to keep current with all of the bad guys and all the different kinds of antigens that might be on their surface, your body is going to produce just lots of lymphocytes that are all trying one potential receptor shape to fit that antigen. So I want you to imagine that you have millions of lymphocyte cells in your body that might look like this, where each cell might have hundreds of thousands of copies of just one receptor shape in an attempt to bind to the antigens that they might come across. So how does that work? Well, through the process of clonal selection then, the idea again is that if you have millions of lymphocytes all trying out one shape, this diagram here is only showing 3 different shape receptors and 3 different lymphocyte cells. But I want you to imagine that there's countless other lymphocytes there all trying out their one shape. Then maybe just by pure chance, there will be an antigen that fits one of the lymphocyte receptors. And so that lymphocyte has been selected, and then the idea is that then that cell will also be triggered through a signaling pathway that's likely activated to do a lot of mitosis and make copies of itself, make copies of the cell with the correct receptor shape to go and deal with that specific threat. So try out random shapes in the hope that one of them will succeed, and then clone that successful lymphocyte. And I'm just trying to summarize here what I just talked about, in case that helps. Here's the basic idea of clonal selection: how lymphocytes find specific receptor shapes to deal with specific pathogen threats and the antigens that are on that pathogen. Alright, so if we found a solution, what we need to talk about next is the different types of lymphocyte cells. Each one of these 3 types that are listed here go through that clonal selection process. So that's sort of common to all 3. What makes them different then is maybe what they do once they've found their solution, their receptor shape, and they've already cloned. What do they then do that helps get rid of the pathogen threat? So we're going to walk through three types. We're going to walk through helper T lymphocytes, cytotoxic, also called killer T lymphocytes, and B lymphocytes. And you really do need to be able to tell me what each one of these guys are doing. So helper T lymphocytes might receive the antigen because they're actually presented the antigen and what it looks like by a phagocyte that maybe previously swallowed it, destroyed it, and there are actually special types of phagocytes. I'm not going to go into specifics about which types of phagocytes do this, but there are certain types of phagocytes that actually display the antigens of the pathogen they just destroyed. And so if presented by a phagocyte wandering around the body, and if a helper T lymphocyte happens to have that receptor shape that fits that antigen, that's going to trigger that particular helper T lymphocyte to clone. And what is its job? Its job is to sort of be like the general of the upcoming army. The helper T lymphocytes themselves don't actually engage the pathogen directly themselves. What they do is they find the other two types of lymphocytes, the cytotoxic T lymphocytes and the B lymphocytes, and they actually further encourage them to do their own cloning. As it turns out, those other two types of lymphocytes have to bind to the antigens successfully, but then they also have to be encouraged by this helper T lymphocyte before they really clone themselves massively. So the helper T is like the general of the army. Just a brief aside, that's why HIV is such a deadly disease. HIV virus actually targets helper T lymphocytes specifically. And if your helper T count becomes low enough, then you have the condition AIDS. What does AIDS stand for? It stands for the acquired immune deficiency syndrome. Your whole acquired immune system becomes deficient if this general is sort of inactive and unable to rally the troops to go fight the disease. So let's go to the foot soldiers of the army then. We're going to kind of see that this war is fought really on two fronts. The cytotoxic T lymphocytes have the job of finding the pathogen that might be inside of any infected body cells and killing those cells. So as it turns out, they're going to bind to the antigen successfully when an infected body cell presents the antigen on its surface. So sometimes when, say, viruses infect body cells, the body cell might actually, as sort of a way of fighting back, presents antigens of the virus on its surface as if to say, hey, I'm sick with this guy. And if the cytotoxic T lymphocyte has the receptor to bind to that antigen on its surface, and the helper T lymphocyte further encourages it to clone itself and divide, then it's going to make an army of cells, and those cells go all around the body cells looking for that antigen. And if they find it, they're going to basically signal the infected cell to do apoptosis, which again means programmed cell death. So let's kill all of the cells that are currently sick with the bad guy, maybe to prevent it from reproducing itself. Sometimes that's called the cell-mediated response. And then there are B cells, B lymphocytes. And B lymphocytes are actually going to encounter the antigen maybe directly from the pathogen itself, because the B cells really have the responsibility of binding to the antigen successfully, just like all lymphocytes. Again, it's going to also perhaps need encouragement from the helper T lymphocyte to truly clone itself massively. But when it does, its job is basically to take the antigen receptors that are in its cell membrane and essentially just release them. So we're going to sort of send all of these receptors to just be free floating in the bloodstream and in other body fluids like the lymph of your body. And when the antigen receptors are sort of ejected from the cell membrane of B lymphocytes, we kind of call them antibodies at that point. And the point of the antibodies is really just to go around the bloodstream as the heart pumps blood around the body, and they're just going to find any of the pathogen that's just sort of free-floating around the body cells, in the blood or in the lymph. And by binding to those pathogens, antibodies can do a variety of different things that eventually help clear the pathogen out of the body fluids. So sometimes they might attract phagocytes that otherwise wouldn't kind of know that they're there. Sometimes the antibodies directly attack the bad guy themselves and cause their destruction. Sometimes antibodies just sort of effectively neutralize a bad guy so that it can't do its what it wants to do to infect the body further. So antibodies just really have the job of going around the body fluids, not infected body cells, but around the body fluids and clearing out the pathogen there. I didn't write this on this slide, but that sometimes is called the humoral response, going around the body's fluids, the body's humors, and clearing out the bad guys there. So this is just a nice little flow chart that kind of summarizes the overall picture. We have three players. We have the helper T cells. The helper T cells, again, have the job of binding to the pathogen, cloning, and they go around and they find the foot soldiers of the specific immune response, the B cells and the killer T cells, and encourage them to clone when they bind successfully. B cells again release antibodies to sweep clean the body fluids. Killer T cells have the job of finding infected body cells and telling them to destroy themselves before they maybe release further copies of the bad guy. So if we kill the infected body cells and if we sweep clean the body fluids, then there really should be no other place for the pathogen to hide. So the last little conversation I want to talk about is that each one of these types of lymphocytes, helper Ts and cytotoxic Ts and B lymphocytes, all make memory versions of themselves. As it turns out, everybody else actively fights the pathogen that's cloned, but they'll quickly, after a few months or maybe even a few weeks, their numbers will start to die back as they don't live very long. So they'll sort of do their job of fighting off the pathogen, but then the army will kind of be reduced to a few again. You might ask why that is, and the broad answer just might be energy savings, right? We don't need to maintain a huge standing army against every pathogen we've ever fought. If we successfully fight off that pathogen, maybe the best idea is to kind of take apart that army again. So as it turns out, some of the cells that are produced through clonal selection, though, are very long-lived. And their job is basically just to float around the bloodstream and have that receptor shape in their cell membranes so that if the pathogen ever returns, we can quickly do clonal selection again without having to generate all these varieties trying to find the right receptor shape. Those memory cells have the right receptor shape, and if that pathogen ever returns with the same antigens, they can just bind and clone and maybe fight off the pathogen before it ever even really causes disease. And that's what this graph is trying to show you. They're saying that maybe if it's the first time you've ever been exposed to a particular pathogen with particular antigens on its surface, then there might be a very long period of time that elapses before you finally start producing a lot of effective cells that fight off the pathogen. And in the meantime, you get sick. You get sick as your body's still trying to figure out the right solution to that particular problem. But perhaps, and sometimes that's called the primary immune response, the first time you've ever been infected with that pathogen. And if we take that same person later, you can see that maybe the antibody and T cell count dies down after weeks. Maybe we've successfully cleared out the pathogen the first time. If we actually inoculate that person with that same pathogen again, you can see that maybe if they're introduced right here, you can see that the time period between that injection time and the time that the lymphocytes build up a strong response is much shorter. Sometimes that's called the secondary immune response because it's the second time we've seen the pathogen, or the memory cells exist there that can quickly clone, and you might not even ever feel sick. You might not even ever know that you're sort of generating this lymphocyte army because they're able to fight off the pathogen before it does the damage that maybe makes you feel sick. And so that's really what the purpose of vaccination is, right? The idea of vaccines is maybe we can inject somebody with a weakened form of a pathogen. Maybe we can effectively show your body what the antigens on a particular bad guy look like, so that your immune system can figure out the right solution and make memory cells for that particular pathogen. So maybe we can inoculate you against, say, the measles virus or the flu virus for a particular year. And maybe you won't get sick with that virus because you already have the memory cells that fight it off before you get sick. So vaccination really has been an important role in promoting the health of populations. There are some people who are concerned with various aspects of vaccination. I will simply say that the biology continues to support the importance of vaccination. You can either get vaccinated yourself and sort of prevent the pathogen from living in your body for some time. Or you can be somebody who chooses not to get vaccinated, and you're really giving the pathogen a chance to live in your body and then in somebody else's body for a short period of time. That really puts us all at risk, because what if the pathogen mutates at some point and changes its antigens a little bit? Well, suddenly my memory cells won't be all that effective. And maybe that new form of the virus can even make me sick, even if I was vaccinated against the old virus. So there really is sort of a public policy debate about how we should think about getting people vaccinated. But I just wanted to make sure you understood the biology of vaccination and the immune system. So, what have we done in this video? We've tried to really talk about the process of clonal selection and how it finds the right solution in the first place. We talked about 3 types of lymphocytes and what they all do once they clone themselves after finding the right receptor shape. And then we tried to talk about the phenomenon of memory of old pathogens and how you don't get sick the second time with the same pathogen.